Professor Ray-Chaudhuri, who was former head of our department, and he was also an eminent scientist and a teacher who has played an immense role in establishing genetics in India. So with that, I also want to welcome our chief guest, Professor Shyam Sundar, our esteemed speaker, Professor B.K. Thelma, and the member of S.P. Ray-Chaudhuri Memorial Foundation, Professor Rajiv Raman, Madhu Tapadia, Dr. Gaurav Pandey, and I also request you all to please come to the dais. Dr. Gaurav, if you can help with.
Children. एक चीज दिक्कत है लेकिन हमें तो ये स्क्रीन दिखानी पड़ेगी ना.
Friends, welcome to this gathering. Last 2 years have been particularly devastating because of the corona infection, and so many activities could not be done. So much so that this series of lectures could not be done for last 2 years. But what was the most saddening part of it was that we lost a number of friends, colleagues, teachers, dear ones, near ones during this period. From our point of view, most notably, we lost one of our colleagues and teacher to many of us, Professor Jaipal Gupta, who was a member of the governing council all the time of this foundation. But he was among the first few students that joined Professor S.P. Ray-Chaudhuri for PhD here. Professor Ray-Chaudhuri put him on the path of a very uncelebrated subject at that point of time, that of Drosophila taxonomy, which Professor J.P. Gupta took it very seriously, and he became an international person of authority in that subject. During this corona infection pandemic, we lost Professor Gupta rather suddenly. He was one of the most self-effacing, least publicity driving person, one of the most humble teachers and an inspiring one. I would request you all to stand for a minute's silence as a token of respect and peace to this soul.
नहीं स्टॉप स्ट्रीमिंग तो हो जाएगा. इसको आगे रहने देते हैं क्योंकि जब पीछे चल जाता है ना गूगल में तो समाओ ये नहीं लेता है. थोड़ी देर में देखता, थोड़ी देर यूट्यूब तो थोड़ी देर बाद ही चलता है ना स्ट्रीमिंग थोड़ा. इसको आगे रहने देते हैं.
Respect. Thank you. With this now we will start the proceedings of today's.
So at our university, we always start an event with the recital of our Kulgeet. So I would ask our PhD student to come, Olivia, Abhijit, Devashruti, Naveen, Anjali, Tanvi, and Sagar to render the Kulgeet. अतीव सुंदर ये सर्व विद्या की राजधानी राजधानी मधुर मनोहर अतीव सुंदर ये सर्व विद्या की राजधानी राजधानी ये तीनों लोकों से न्यारी काशी सुज्ञान धर्म और सत्य राशि बसी है गंगा के रम्य तट पर ये सर्व विद्या की राजधानी राजधानी नए नहीं है ये ईट पत्थर है बेशर सुंदर मंदिर ये सर्व सृष्टि की राजधानी राजधानी यहाँ की है ये पवित्र शिक्षा कि सत्य पहले फिर आत्मरक्षा बिके हरिश्चंद्र थे यही पर ये सत्य शिक्षा की राजधानी राजधानी वेद ईश्वर की सत्यवाणी बने जिन्हें पढ़ के ब्रह्म ज्ञानी ते व्यास जी ने रचे यहीं पर ये ब्रह्म विद्या की राजधानी राजधानी वो मुक्ति पद को बिताने वाले सुधर्म पथ पर चलाने वाले यही फले पूरे बुद्ध शंकर ये राज ऋषियों की राजधानी राजधानी सुरम्य धाराएं वरुणासी नहाए जिनमें कबीर तुलसी भला हो कविता का क्यों ना कर ये वाद विद्या की राजधानी राजधानी विविध कला अर्थ शास्त्र गायन गणित खनिज औषधि रसायन प्रतीचि प्राची का मेल सुंदर ये विश्वविद्या की राजधानी राजधानी यमाल जी की देशभक्ति ये उनका साहस ये उनकी शक्ति यमाल जी की देशभक्ति ये उनका साहस ये उनकी शक्ति प्रकट हुई है नवीन होकर ये कर्मवीरों की राजधानी राजधानी मधुर मनोहर अतीव सुंदर ये सर्व विद्या की राजधानी राजधानी मधुर राजधानी राजधानी मुझे शायद एक बार बोलना चाहिए था क्योंकि यहाँ बहुत सारे नए स्टूडेंट भी बैठे हुए हैं। कुलगीत के बाद हम लोग तालियां नहीं बजाते। So with that, I would ask Olivia to please come and present the floral bouquet to our chief guest, Professor Shyam Sundar. Is from IMS BHU. Thank you, Olivia. Now I request Devashruti to please come and present the flowers to our speaker, Professor B.K. Thelma. Thank you, Devashruti. So now I request Professor Rajiv Raman, President of Professor S.P. Ray-Chaudhuri Foundation, for the welcome address. So please.
Professor Shyam Sundar, the chief guest of this evening, Professor B.K. Thelma, the speaker. Am I audible? Does it need to be? Professor Madhu Tapadia and Professor Gaurav Pandey on the dais. Professor Anil Tripathi, the director of Institute of Science.
So you're audible, not visible.
I'm audible, not visible. Is it
Yes.
visible?
Yes, sir.
Okay. Rachel, I don't know what ways it can be made more visible.
Yes. Now you're visible too.
Okay.
Thank
So,
you.
so I'd say it's a great privilege for me to be standing here before you to welcome such an august gathering here, and such distinguished people on the dais and in front of me, and a very big population of students, which is what is the soul and essence of any university, any academic institution, and of course, many more colleagues here. So, hearty welcome to this function, which we try to do every year in honor of Professor S.P. Ray-Chaudhuri. Those of you sitting in this room, which has the name Professor S.P. Ray-Chaudhuri Hall, would understand that we are talking of the same Professor S.P. Ray-Chaudhuri, who was a doyen amongst the geneticists of this country, who did his MSc in zoology from Calcutta University and then went to England to do his PhD. To begin with, he went to do his PhD on silkworm genetics with the FA crew. But as Providence would have it, Second World War broke out, and Hermann Joseph Muller, who later got Nobel Prize, had to escape from Russia and come to Edinburgh. And when he was admitting students for PhD, S.P. Ray-Chaudhuri was one of those who joined him. And he worked on what was called as the extremely low radiation and its effect on genetic material, a matter which has been discussed for all these years with great interest, and also a matter of great contradictions and so on. So, having come back from there, Professor Ray-Chaudhuri was serving in Calcutta University. But as you know, Drosophila genetics needs kind of a setup. It needs air-conditioned rooms and all that. In Second World War and thereafter, the country was in a condition that it was not really easy to do that. So he started working on cytogenetics of grasshopper to begin with, on radiation genetics of grasshopper. And that is how, in certain ways, Professor Ray-Chaudhuri is the pioneer of animal research in genetics and cytogenetics. There were good researches done on plant breeding and plant researches. There were good research being done on livestock researches and breeding and so on. But a systematic research on genetic principles and its teaching goes pretty much to the credit of Professor Ray-Chaudhuri, who in 1960 came here. Actually, it's interesting to know that he started his Drosophila genetics because of the courtesy of J.V.S. Haldane, who came to Calcutta University and found out that Professor Ray-Chaudhuri, who at that time was an assistant lecturer, was not able to do genetics because he could not have a lab. Haldane was an important person. He went back to England, wrote to Nehru, the Prime Minister then, and said, I want to come to India and start genetics, and why don't you get me a lab made? I want to come to Calcutta. A letter went to the Vice Chancellor of Calcutta University saying that a lab should be made for Haldane. And when the lab was made ready, Haldane came and told Ray-Chaudhuri, lab is for you. That's how Drosophila genetics research in India started. In 1960, he came to this department, and Professor Ray-Chaudhuri his merit lies not merely in doing very good research and being an extremely good teacher, but making its life mission to see that good science is taught and researched upon throughout the country, and genetics, having been the subject in the 1950s, genetic material having been discovered, became a topic which needed to be taught and worked upon all over. And he worked assiduously to see that not only in BHU, but everywhere else in India, this subject came up. And this will not be the only subject which is necessary to be propagated, but there would be subjects like physiology, developmental biology, animal behavior, ecology and all. All these need to come together to be able to make animal science a vibrant subject that will contribute to science in a big way. And in so doing, perhaps one message that he gave to all of his students was doing good science, having a scientific temper, and spreading science to as far as you can. Professor Ray-Chaudhuri passed away in 1994, and when he passed away, his students, colleagues, well-wishers, friends, and his family members, they all thought it proper to have a foundation in his name devoted to expanding science to the general public. That is exactly the objective of this foundation, which was initiated by Professor Tikaram Sharma, who was the first president of the foundation, and we have been trying to do this to the best of our capacity. I'm so glad that we are all here again for the 22nd lecture. The first lecture was given in 1994, the year Professor Ray-Chaudhuri passed away, by Susumu Ono. And thereafter, we have had at least 20 more lectures. The last lecture was given in 2019, shortly before the pandemic hit us hard. That was given by Professor Gagandeep Kang. And it was told that the foundation does not get lady scientists. So Gagandeep Kang was the first woman scientist speaker. We are very fortunate that now we have the second speaker here, Professor B.K. Thelma, who is a lady scientist. But gender does not matter in science, so it doesn't make a difference. It is a good science which matters, and I'm sure you are up for a feast. Hearty welcome to all of you. Thank you very much for being here. Thank you.
Thank you so much, Professor Raman, for telling us about the passion and perseverance of Professor Ray-Chaudhuri. So now I would like to invite Professor Madhu Taparia, to the secretary of Professor S.P. Ray-Chaudhuri Foundation, to introduce our speaker.
Thank you, Richard. Professor Shyam Sundar from IMS, chief guest of today's talk, Professor B.K. Thelma, Professor R. Raman, Dr. Gaurav Pandey, Professor Anil Tripathi, our director, all distinguished faculty members sitting here, colleagues, students, friends, everyone. It's my proud privilege, I consider proud privilege to be introducing one of the most renowned women scientists, as Professor Raman mentioned, in India, who is known for her consistent and original contributions in the field of human genetics. And she's on a way to transform medicine into precision medicine, which you will hear about when she gives her talk. But what I would like to emphasize over here, especially the students who are sitting over here, that if you have a dream and if you follow it, then with conviction, then you will not be disappointed at the long run. That you will realize when Professor Thelma talks about her work. She was born in Kurg in Karnataka in a large joint family, and the first lessons that she learned was family bonding and taking care. And this family wellness was so much imbibed into her that she wanted to become a doctor when she did her school. However, but her father wanted her to become an IAS officer or take up maths. But then she could not get into medical school because her age did not permit her. She was of lower age. And so, and this was, I think, because fate had something else in store for her. So she joined BSc, and then she did a post-graduation with Cytogenetics major. And again, as I learned, this was not her first choice. So after her master's, she was looking for a place where she can pursue her dream of working on biomedical field. So she joined Professor SRV Rao in Delhi University. But there also, again, she was not directly working with the biomedical line. She worked on the cytogenetics of Nesokia mole rat. But her dreams got wings when she joined Professor Hermann Jacob Müller lab in Switzerland for a postdoctoral position. And that was in a hospital setting. So she was finally in a place where she actually wanted to be in the first place. And she, after her postdoctoral tenure, she came back to India. She joined as pool officer, and she became a permanent faculty in University of Delhi in 1987. And there she first worked on the sensitive sites of X chromosome, the fragile sites, again in mole rat. And it was later in 90s that she set up her own laboratory. She set up a human genetics laboratory unit in South Campus of Delhi University and started working on the Fragile X syndrome of human. And hers was the first lab which started molecular diagnostics on Fragile X syndrome, and then with the schizophrenia, Parkinson's, inflammatory bowel diseases, and so on. And at the same time, she got a major human genetics project from the DBT, and successful completion of one project led to another, to another, to another. So as she climbed up the rungs of a ladder, honors and awards followed. She is member of all the three academies, and she got JC Bose National Award. She also won Hora Award and Sir Hora Award. And last year she received a Karnataka State Sena Rao Award for science stream, and but she got the award this year. So what I'm saying is that she at present has been able to fulfill what she had wanted to do when she was a young child. So if you follow your dreams, I think there's no way that you will be stopped. There'll be hurdles, but you have to overcome it. Apart from being a good scientist, she's also a very good teacher, a mentor, and she is very friendly with the students. And so, with these few words, now I request Professor Thelma, please deliver her talk.
स्टॉप दिखाई मैं करवाती हूँ इंटायर स्क्रीन कीजिए करो देख सकते हैं पीछे वाला ऑन कर
Ask them to put on the light, no? Okay. Okay, now what do I do to just use the keep? Okay, I have to keep doing down, no? The sun kind of stuck there. Okay. Give allow. Not to worry. Not to worry. You just have to use the mouse, right? No, I mean just to put this down. Okay, all right. You want to have the light on first or no? That's okay. You want this also and this?
No, ma'am, I think that is
I think this. Yeah. Yeah, maybe for a couple of minutes. Yeah. Okay. A very good afternoon to all of you. At the outset, I must express my gratitude for having me here amidst all kinds of difficulties. And I think it's a wonderful opportunity for me to share the fact that, Professor Ray-Chaudhuri was a very special person. I had no direct interactions with him, but through his mentees, first generation, second generation, and third generation, I have known what a master he was. And therefore, I think it's a wonderful opportunity to pay respects through this talk. So thank you very much, Professor Raman and the team, to have me here. And secondly, I'd like to say thank you to Madhu. You conveyed the message that I want to put across, especially to the students, that you have a dream, you have to have the drive to pursue it. It has to be a passionate one, and you will reach where you are. That's really the final message that I want to give, even before I start. I also know that the audience is mixed, so I've made it really, really very basic. So please pardon me, all the scientists here who know the subject, you'll have to bear with me because I made it very simple with very minimal technical details. But message is that, one needs to appreciate where all genome research is going to achieve the larger aim of conquering genetic diseases. That's really the message that I want to make. So if there's a little light here, that will actually help me. Okay, so it's very that this picture may not be very interactive because of constraint. Essentially, what it represents is a lot of variations that you see. It doesn't matter man, mouse, whichever species. Unlike this is genetic variations. That is what determines the group of us here are so different from each other, except I don't see monozygotic twins, where you would have similarity in the phenotype, maybe 99.9%. But this is what is really the underlying factor. And addressing or identifying variations which have implications for health and disease, main crux. So having said health and disease, if we talk or restrict ourselves to human genetic disorders, broadly it is chromosomal disorders, which contribute about 6 to 8%, monogenic or single gene disorders, which are pretty common, which most of you know. You talk about thalassemia, sickle cell anemia, and so on, which contribute another 6 to 8%, and complex traits, which are over 60%. And common complex traits are typically lifestyle disorders and type 2 diabetes, hypertension, Parkinson's disease, rheumatoid arthritis, a whole lot of these fall under these complex disorders. So to put everybody on the same stage, I will actually kind of go through with few basics to start with so that I don't lose anybody at the end of the talk. So chromosomal disorders are very standard, known for the longest period of time. All that you need to do is make chromosomes from the blood cultures, and you can identify both numerical aberrations as you see. Is there a pointer that I can use on this? If you can show me. Otherwise, you can give me a stick pointer. That is okay, all right. Okay, so here you have 3 chromosomes of 21, which makes it a Down syndrome. Everyone in this room could have seen a mongoloid child. Okay, very straightforward, simple diagnostic procedure. And it's also easy that you can do a few more molecular analysis to identify not just the numerical aberration but also the structural aberration of a translocation that you can see here with two different colors exchanged on the two chromosomes. These have major diagnostic implications which has been in practice ever since the 1960s and the 70s. This is one technique all through in molecular diagnostics which has stood the test of time and which continues to be there. Yet, unfortunately, most people still end up having a Down syndrome child because the access or awareness among the public is limited. Prenatal diagnosis is possible, medical termination of pregnancy is possible when it comes to these simple chromosomal disorders. But when you come to single gene disorders, it's a little more different. We're talking about one gene, one disorder, but it's one mutation. Let's say thalassemia, you're talking about one specific mutation, which is going to be there in all the members, wherever it's present. Parents are carriers, but you may have an affected child or absolutely normal child or another child like the parent as a carrier. Unlike that, the complex disorders are a lot more difficult because you have more than one gene. If you ask me how many genes for type 2 diabetes, the answer is I don't know. Not just me, I don't know. Nobody knows how many genes contribute to any of these common complex traits. Together with that, it is the environment or the lifestyle which contributes or which makes the susceptibility even more stronger and expresses the disease. So right there you will know addressing genetics of single gene disorders is far more easier than complex disorders. So if I have to tell you upfront what the message is, that we have no answers to anything as far as complex traits are concerned at this point in time, while a lot of progress has been there in single gene disorders. So let's quickly look at what's happened over the last 6 to 7 decades. Chromosomes, as I said, started in 1960s, continues to be in practice and in clinical use at this moment. Then you had recombinant DNA era, which dominated, starting with cutting of the DNA, ligating the DNA, cloning of the DNA, and a lot of different things. The most dramatic shift in genomic advance happened in the 1990s when the Human Genome Project came. Most of you would have heard about the Human Genome Project, which simply aimed at sequencing the entire human genome. I'm talking about 1987 through 2000, where it was aimed, and following which there was the fallout of the Human Genome Project led to analysis at a larger scale for common complex lifestyle disorders. And today, everybody is talking about next generation sequencing. In other words, from a project mode, it's actually become a routine clinical practice where you just get everybody sequenced with the hope you identify the variant. Okay, and there were expectations. What were the expectations? 2010, I mean, this is an old slide and I always like to keep it because it just said in 2010, they said there'll be lots of tests which are available already for some conditions, gene functions will be known, etc. 2020, they said there'll be gene-based designer drugs because they thought a lot of genes will be identified, variants will be identified, there will be interventions through various gene therapies, but sadly, that hasn't happened. Cancer therapy made some progress, but not all that is desired because newer things happen. The prescription testing of pharmacogenetics again was expected to be all in practice, but we know it is not there and there is a lot of struggle on a lot of things. So what was expected in a few years from now? Comprehensive genome-based healthcare and understanding everything of a human cell or the disease condition through a computer model. And major anti-technology movements, just to put it in the context, you talk about the CRISPR today, right? So that's CRISPR-Cas9 genome editing to make designer babies, for example, is what we're, other than the regular gene therapy and cell therapy and other things. Then whether humans will take their own evolution into hand. So this is what expected by 2030. Let's see at the end of the talk whether we're anywhere near that or some progress has been made. So I now switch gears from classical cytogenetics of chromosomal testing to what we refer to as the P4 medicine. Ultimately, what is important? For you to be able to eliminate a genetic disease, you must know what the cause is. In other words, there has to be a genetic diagnosis for it, DNA-based diagnostics. If you know there is a particular mutation, you can work on that. Look at the family which has a particular disease which is running. Make sure you can give them prenatal diagnosis or counseling, appropriate counseling, and you can prevent it. If you can't prevent, you would like to have cure. Standard one, prevention is better than cure. So if you can't, the last resort is treatment. So this is where we are in medical. That's a central dogma in medical biology now. Diagnosis, prevention, cure, and treatment. And this essentially, combined with the genome analysis tools, has taken us to what is being referred to as P4 medicine, standing for predictive, preventive, personalized, and participatory. If a family is not willing to go for a testing and make sure that the disease is addressed or eliminated from the family, there is nothing you can do. But that's human rights, right? So you have to have the participatory nature in all of this P4 medicine. I normally put these questions at the end, but again, as I said, because of the mixed audience, I want to ask the questions right up front. Do we know all the genes in the genome? Despite 30 years now of the Human Genome Project, answer is no. Do we know all the disease-causing genes and variants? Answer is no. Do we have genetic testing for all diseases? Answer is no. And some of us get a headache. I'm okay with one Dispirine. Somebody says Dispirine doesn't act. Somebody says I need 2 at a time. Each one of us respond differently to drugs. That's again got a genetic basis. Do we have biomarkers, early disease prediction? When you say genetic marker, that's also a biomarker, but do we have any other kind of early biomarkers for disease prediction? Because early diagnosis is very important for prevention. And lot of structural biology, lot of protein structures are being solved. Are we going to get new therapeutic molecules because we understand? Are we going to address all the issues? So these are a whole lot of which the answer at this point, no matter what the advances in technology is, the answer is no at this point. I like to add 2 more because we are going to talk about examples from Indian population. Therefore, I want to ask, why can't we use Western data? Why is it that we need to kind of spend time and money to look at populations and try and do it with Indian samples? And why can't we use animals for all our experiments? So I will start addressing the last 2 questions first before I go on to the others. So if you look at this, it's a man and a mouse, chromosomes, just painted chromosomes. Forget about anything, just look at what is there in terms of color code. So I'm going to the details, but all is that a lot of chromosomal change and of the homosexual which has come. So while there is a lot of similarity in the of, let's say, for example, you have a whole lot of differences because your gene expression is dependent on the organization of the genes and the contextual position. And that is what really makes the difference in terms of complexity and developmental differences between, say, for example, the mouse and the man. And this is true for everything else. So if you look at it, those of you who are here who would have already noticed, the X chromosome and the Y chromosome is the one which is maintained pretty much unchanged. In other words, the X and the Y chromosome are highly conserved, while a lot of reshuffling has happened all over. Okay, a cartoon just to make you laugh. Okay, this is the kind of similarity we talk between organisms and humans. Okay, and yet there is no way you can substitute human sample or patient sample research with any, because in vivo biology is really the paradigm. Coming to the second question that I asked, why should we really look at specific populations? Why can't I use a lot of Western data? They're 20 years ahead of us in every aspect of genome analysis. But that's because we have, Indian population, there's a lot of inbreeding, very large families are available. Because of inbreeding, you have a greater chance of having autosomal recessive diseases higher. You have various socio-cultural practices. Your environment is different, geography is different, food habits are different. So all that also makes a difference. And a genetically or a ethnically distinct population like the Indian population is a very, very ideal material for us to contribute to actual discovery science or discovery genomics in the global context. Of course, we have lots of difficulties. We don't have epidemiological data, we have lack of awareness, many, many things. But despite all of that, we definitely can contribute notably to understanding genetics and contributing to discovery to start with. Just again to highlight, this is what the population structure is. Don't worry about anything. These are different populations, the African population, the Chinese and the Japanese, but very, very specific overlap. The lowest one is the Caucasian there, which is blue. And then you have the Indians. Don't worry about the larger size. That's simply because larger number of individuals have been plotted. You have green, which is North Indian, and orange, which is Gujaratis in Houston, which are the HapMap, genomes. Just to show that Indians are indeed genetically distinct. And even if you want to kind of look at the difference between North Indians and South Indians, you can see a little bit of a difference, but that is together in a cluster there and so on. That's just to give you a message that it is worthwhile for you to look at the genetic distinctness of the Indian population and to utilize it meaningfully for discovery. This is again just the same message which we are going to put across. So transethnic studies are extremely useful. So what I'm going to do in the next few minutes is I'll tell you broadly where we are, starting with the Human Genome Projects. I like to say the information that we got on the variations in the genome, which is what is making us different from each other, from each one of you, different from your sibling, and so on, is because of the variants which are there in the genome, which I like to call as spelling mistakes with some kind of severity, differences which may be there. And then how do we use this patient information for identifying new disease genes? In the next section, I will tell you how difficult it is to reach complex disorders and whether combining Ayurveda together with genomic tools will actually help. And finally, I will close with an example for Science for Society. Again, this is for students. Please forgive the rest of you. DNA double helical molecule packaged beautifully into a chromosome. The Human Genome Project wanted to identify all of the sequences in the human genome. That's a karyotype. And they said short-term goal was to do the sequencing and annotation and characterizing the gene. Long-term, they said, was gene therapy, pharmacogenomics, and basic research. Alas, they said they thought that Human Genome Project, everything will be identified. Old age diseases will be all conquered because you would know the genes. But what we ended up with a very, very unexciting set of sequences, which till today largely remains ununderstood or ill understood. So it's exactly like taking a language textbook, take off all the punctuations, capitals, etc., and try what it reads like. To make sense of a paragraph, I'm giving you an example. Sadly, I don't have time to ask you to read out and make words from here, but some of you would have already seen some words which can be easily identified, while you will struggle to make sense of the rest of it. But if this is what is given to you, you know what it means. This is exactly where we are even today with a large part of non-coding sequences in the human genome, where we have difficulty in understanding what they are doing or how they are regulating gene expression. So, when I said spelling mistakes, these are because of variations. A car and a cat mean the same thing, okay? But there is a change. But if I said I'll take a bar, one letter changed in the word, but it means something different and probably meaningless. I will take a jar is not what you started off with at all. The same thing happens in translation. A change in your base changes the amino acid and therefore the protein, and that is what the meaning of the spelling mistakes and the consequences. It could be a car and a cat situation where it hardly matters, or it could really be a mistake which changes the protein, resulting in a dysfunctional protein or an aberrant protein, resulting in disease. so on. You can have different ways of spelling and so on and so forth. So, classical example is sickle anemia. One base change makes it a sickle, and that's an example. So, if we summarize this part and say, today we know only this portion which is marked in red is what is really the coding sequence or the protein coding portion, which is less than 2% in the genome. The rest of it is a variety of other sequences, and these changes are referred to as single nucleotide polymorphisms and SNPs in short. In other words, there is a little distinction between a word mutation and a variant or an SNP, because mutation is something which is already proven to have a pathological consequence, whereas when you call something as a SNP, it may or may not have a major effect. Okay, so this sums up what really the changes in each one of us do, and why each one has a different susceptibility to a disease, etc. So it's essentially saying it's not important. A play is therefore an apposite metaphor of the genome, in which the principal interest lies not in the word or letters, but in the way it is performed and brought to life on the biological stage, emphasizing the paradigm of in vivo biology or the importance of in vivo biology. And Human Genome Project and sequencing resulted in a variety of omics-related activities, and based on this, so this is the kind of a time when I started research with my own independent project. So broadly, discovery genomics was the primary interest of the lab, and translational medicine, new drug molecules, is the ultimate aim. But if you really kind of look at it, there are various ways that you can do. You can look at complex traits, you can look at single gene traits or Mendelian traits, you can do variety of analysis of the gut microbiome or iogenomics or functional genomics, understanding disease biology and disease risk prediction being the main aim. At the end of the day, you don't want it to be just ivory tower research. You really want translation to the extent possible, and to develop newer therapeutics, because we still are very, very far behind as far as new molecules are concerned. With that, I will switch gears and tell you a few examples. When I said India is rich in genetic material, this is what I mean. Here is a family, and since most of you may be genetics, you may be learning genetics in your master's or you've learned it in undergrad. Here is a pedigree where every black filled box is an affected individual. 4 generations, 1, 2, 3, 4, 4 generations of the family, multiple boys affected. Clearly, it is an X-linked condition. It came to the lab thinking it was a Fragile X syndrome, because that's an X-linked condition. But we were lucky, it was not Fragile X, but we turned out using the next generation sequencing technology and the really large informative family, we were able to identify a novel variant in a gene called MID2. Variety of techniques, that's not important to know, but MID2 was not discovered before by any Western lab. The family of this kind helps you to identify novel disease causal genes. Okay, so this is way back. It's almost 8 years since we got. This is the first one that we had identified. Similarly, we have another one. You can imagine how unfortunate this family is. 5 children, 4 boys and 1 girl. All 4 boys affected with what is called a Sickle syndrome. Okay. They continue to have children. In the West, probably doesn't happen. 1 child affected with 1 gross phenotype abnormal, they would stop there. But here, in anticipation that the next child is going to be normal, you end up. And this is what happens. Very rich resource for people like us to work on, but very sad for the family. 10 years to get at the gene which is responsible for this, because this is not one of those classical ones where the gene was but the gene happened to be in a regulatory region. It happened to be a deep intronic variant, which was a leaky splice variant, which resulted in aberrant transcripts leading to disease in this family. So this manuscript is finally in revision. So it takes a while, despite material, despite whatever, because regulatory variants in Mendelian disorders are just emerging. Otherwise, you're normally searching only for protein coding variants. Parkinson's is yet another example where many genes are known for familial Parkinson's. I'm not talking about the age-related Parkinson's, where it is a lot of number of genes which contribute, but I'm talking about a familial one. Here is a family of 8 children, 3 of whom are affected. I have a lovely video, but because of the time I won't show you. But 2 boys and 1 girl affected. We went ahead, did the analysis using the next generation sequencing technology and comparing the healthy versus the affected, we were able to identify the gene. I like to just point out with the model here, maybe Lakshmi is in the audience, is one of the students who's now working in BHU with Dr. Marshumi, who contributed to some of these gene discoveries. So here marked in red are 3 genes from 3 different pathways that we have identified for Parkinson's disease. Don't need to go into the details, but just to tell you, so much is already known, but so much remains to be known, and every is a very important material to be able to contribute to disease causal genes, which means important for the family immediately, but also important for your diagnostic panel in the larger context. So these are all the genetic contributions, the contributions in terms of gene discovery. And when you discover a gene, the next step is to go back and do functional characterization, because you will not be able to publish a good paper if you don't characterize. Functional characterization is not a one-step process. It is a highly integrated approach using everything. I might have an example later on that I'll show you, but functional characterization of these genes will give you some clue. It will give you a clue to the biology underlying the disease. It might, if you're lucky, give you a clue whether it is a likely therapeutic target. So from single gene disorders, which apparently now seems straightforward because next generation sequencing tools are there, you have the families, you are able to identify new genes. What about complex traits? I already told you more than 1 gene, we don't even know how many, and there are many variants which contribute. They're all genes of minor effect. Many genes of minor effect. But just to tell you, instead of a plus or a minus, all that happens here is all of us have the variant, okay, in 1 gene. But in another 10 genes, some of us have the variant, some of us don't have the variants. So the permutation and combination of the number of variants is what pushes somebody in the audience to develop diabetes, to develop rheumatoid arthritis, while the others, with maybe several of the variants contained in their genome, still can escape developing the disease. So these are called as just association studies, where I will just count the black as the protective or the normal wild type allele. The red ones are the predisposing ones. So obviously, when I'm looking at this, I'm simply saying that a healthy individual will have a fewer of the predisposing alleles, whereas a patient group will have a lot more of that red color. So you do statistics and try and identify the gene. So instead of just identifying a variant, you look at the SNPs, you do statistical analysis, and you are referring to this in a genome-wide fashion. That is what is called as genome-wide association study. We were fortunate to be able to do this for 2 diseases, rheumatoid arthritis and ulcerative colitis, and we actually identified new disease susceptibility genes not reported anywhere in the West. Okay, so this is just to say that there are many genes which are known for many complex disorders, lot of overlap. One color can be seen for many diseases, and they're involved, but transethnic studies are important for you to be able to identify. I will illustrate the next 2 sections with rheumatoid arthritis as an example, where we did a genome-wide association study using rheumatoid arthritis. We identified a new gene, which is called as ARL15, and functional characterization using multiple omics tools. We did transcriptomics with the knockdown of the gene, overexpression of the gene. We did proteomics, we did pull down, all characterize the gene because it was something which was a non-Chile gene, which was special for rheumatoid arthritis. We could go ahead and look at omics finding that, and it was a potential drug target. So it's on a very simplistic model that we put this at. The presence of a variant of this gene will increase the levels of the inflammatory molecules. So in other words, if we use an inhibitor of this gene, we should be able to suppress the inflammation. Simply said, that was the model that we went for. And I won't go into the details, but we've been extremely lucky that we did CIA model, that is the legend-induced arthritis model of mice, and we have been able to show that indeed using the monoclonal antibodies to start with and small molecule which was designed against this particular protein, we have been able to show that there is a significant efficacy of this molecule. Don't worry about the top, which the mouth pause, because that's what you will look for, the swelling and the joint swelling, etc. Panel. This is of the bones to tell you whether your treatment really been treated. You can see all the deformities which is captured in the micro CT. Experiment. Antibody that we used against the target gene, that is our novel Jiva gene that we identified under patenting process. And I'd just like to say that that's actually an example where we talk about a gene discovery to a new drug target. So monoclonal antibodies are not the best way. You like to develop new small molecules. So that is what the lab along with collaborators have been doing, to develop new molecules or synthesize new molecules to see their efficacy, etc. So that's a classical example where we have been just lucky. Over a 10-year period, we have been able to just reach from the gene to the drug target. Now, having said that, this is all hunky-dory pip-pip because I've just told you things which are all success stories. But now you can turn around and ask me, if so much is done, why is it that you can't predict type 2 diabetes or hypertension? Why are people suffering? Common diseases, I'm sure in the audience, there are at least 5 people in the audience who would be having 1 or 2 of these conditions. Why is it that we don't have any interventions, don't have biomarkers? And that's because if you have gone to a clinic, everybody who is a type 2 diabetes is put into a diabetology clinic and they're all marked as just type 2 diabetes. Okay? In principle, each one of us are different. Our genetic makeup is different. Our clinical symptoms are different. So there is so much of heterogeneity which is there, both clinically and genetically, which is what has to be addressed and which is what our contemporary modern medicine does not permit us to do in subgrouping. And that's where we have used Ayurveda, which is, we take an earliest example of people medicine, because if those of you have an experience with Ayurveda know, no 2 patients are ever treated with the same medicine. No 2 patients will ever be having identical kind of conditions or constitution. So we decided to look at this powerful tool of what we call as de-phenotyping. In other words, you do a constitution-based analysis and subgrouping of individuals, combine it with the utmost or the most contemporarily available genomic tool, and then try to do association studies to pick up genes of interest. So I won't go into details, but it's a holistic Indian system, as all of you know. It is taking care of the gene-environment interactions. There are basically 3 constitutions that you talk about, the Vata, Pitta, and the Kapha. Each one has distinct characteristics, and the emphasis in Ayurveda is really not on addressing what the symptom is, to really get at the cause of the disease. And therefore, the treatment is going to be far more effective because you are addressing or identifying cause just like what we said in single gene disorders. But if we say that, it's a lot of things which are already recorded. It is you are what you eat. In other words, you develop diseases depending on your food habits. In other words, if you control your diet and if you eat the right things, you definitely can avoid getting that particular disease, or you can help yourself maintain to whatever is possible. So it's very, very important for you to identify the person. So this is really what you have by way of a diagrammatic representation. Here you have a set of people, which is what allopathy does, where you have all of them put in the left category, which is here, everybody put together. In reality, each one of us in this room fall into not just 3 categories that I've shown here, but you into different groups. There are a total of 7 subgroups, but predominantly we talk about 3 subgroups. And therefore, if you look at the predominant 3 subgroups here, and then you go ahead and do the analysis, we can identify of some of these common lifestyle disorders. This is just to show that the elements are different in the Vata, Pitta and the Kapha Prakriti groups, and the recommendation is different. For example, Vata group is supposed to be more susceptible to neurological disorders, whereas the Pitta group is more prone to digestive disorders, and Kapha group has more of congestive disorders and so on. So you definitely have a variety of differences amongst each one of us, but which is never addressed when you go and go to an allopathic clinic or to modern medicine. So it is essential now to have a scientific validation of this beautiful Indian system of medicine. And if we are able to combine genomic tools, we think it will give us a lot of answers. And we have done this using rheumatoid arthritis as a proof of concept, where don't worry about this, case and controls. You just use cases and controls which match properties and go back to a genome-wide association study. And not to share the results, but just to tell you that we have identified pathways such as apoptotic pathway in the synovial fibroblast, which is the major target cell type in rheumatoid arthritis, not published in any of the Jivas, the Caucasian studies. 108 loci are already known. This is not one of the 108. Or we have several other genes, including the presentation gene, the antigen presentation gene, which has been captured in the Vata group, and so on. So this is a revision at the moment, but it is just telling you the power of using deep phenotyping, which is an inherent gift to Ayurveda, what is available to the Indian community. But now I know more than Indians, a lot of the Western labs which are actually wanting to use this deep phenotyping method. So, what really listening required to have robust tools, biomarkers, where disease prediction should be possible, where disease course prediction, because somebody has a very slow progression, somebody has a speedy progression of disease. So you should be able to have markers for all of these things. That is what we talk about, biomarkers for different phenotypes. And of course, there is a big need for new therapeutics. Repurposing of the drugs. A drug which is known for a different condition is now being repurposed because we understand that there's a lot of shared biology. So drug repurposing is becoming again a big thing, and alternate and complementary therapy is again something which is gaining a lot of importance. So we are talking about a multidisciplinary integrative effort if we have to really address complex traits. Genetics alone is not going to address that. We need to have a multidisciplinary approach. And so to achieve the goals of P4 medicine, particularly in the context of lifestyle disorders, which are the killers today, this is what we have been trying to do in the if we look at multiple omes. In other words, the genome is important, the phenome is important, epigenome is certainly important, but epigenome has a slight limitation because I may not always get the target tissue, and epigenome or epigenetic profile is targets. Genome doesn't matter from where you take the DNA. Okay, whereas when you talk about epigenome, the context or the tissue is extremely important. And on the other hand, you have the gut microbiome, which is turning out very, very important in today's context. And there's a metabolome, there's proteome, a variety of other things. Can you handle all of this just like what you would do with a straightforward statistical analysis? No. That's where your artificial intelligence and machine learning tools are coming in a big way to look at this omics data, and hopefully in the next years, one should be able to have some amount of prediction and prevention of some of these conditions. And that's what we refer to in a loose way as precision medicine, because basically you're trying to say, can I know which is the major most gene which is conferring susceptibility to rheumatoid arthritis in a patient? It's not going to be all of the 10, 20 genes. It may be one which is a major one. Maybe inflammatory marker is not really important there, something else is important depending on your prakriti. Okay? If you rectify your eating habit, half your problems may get solved. This is what the gut microbiome does. When you talk about therapeutic interventions, you talk about a lot of cleansing of the system, cleansing of the digestive system, and repopulating with the right bugs. This is the way one is actually looking at it. So combination of AI ML methods with all of the omics data is really the way which is expected to be meaningful when we talk about the complex traits. So the message is that these are things, it's not fancy science, it is really something which can be practiced by everybody. It is really a common man's science. It's just that you need to be open, you need to appreciate, you need to be able to have some discipline to say, look, I need to help myself. Because no matter what, people are getting more and more ill with common lifestyle disorders. I have not even in this entire talk referred to cancers, because that's a different ballgame altogether. But what I'm trying to say is that a lot of it depends on what you do. That's why you are what you eat, is what I started off by telling you. And let thy food be your medicine and your medicine thy food. Said many, many, many hundreds of years ago by Hippocrates, and that's what we should do. So if we are able to combine some of these things effectively, this thing which was earlier talked about only for pre-prescription testing before a person is actually administered a drug, this can become a routine practice. So you can actually have your genetic profile, which will say what are your disease susceptibilities, what are the conditions that will actually help you, which are the drugs that you will respond to, which are the drugs that you will not respond to, etc., which is going to be a real feasibility. Last part, I hope I'm not run out of time, but just to tell you, Science for Society, all this was research, which is very, very exciting, which is new every day. Newer tools come, newer thoughts come, newer ideas come. So you're constantly, thrilled with what you do in a lab. But how much of it reaches a common man? Okay? Almost all of you here would have heard of, let's say, a common condition like phenylketonuria, right? All of you have heard of this condition, which is an autosomal recessive disorder, an inborn error of metabolism, one mutation in one gene passed on from the two parents who are apparently quite healthy, but the unlucky child gets the two bad copies and the child gets phenylketonuria. Something which is so avoidable and preventable and treatable. Okay? Yet we are not anywhere in doing anything. So new screening for inborn errors of metabolism is something which is addressing the condition of metabolism. Over 600 of them out of the 20,000 odd genes that we talk about in the human genome, about 600 of them contribute are testable. Over 50 of them are treatable. Are expensive or inexpensive, but they are treatable. Most important, they are preventable. That's the message that I want to give across to you. So, in an effort, so every child does deserve a healthy life, and all that this takes is that when a baby is born, it doesn't matter to which parent, any history of disease, otherwise it's not important. All that you need to do is take a heel prick, from the newborn, 24 hours after the baby is born and after the baby has had its first feed. Because you're doing the metabolic profiling. You're not doing the prenatal diagnosis in the mother, but you're doing a metabolic profile of the child. So after the first 24 hours of enough, you do a metabolic profiling for a few common conditions which can be tested, can be treated, and which can be prevented. Okay. So all this I wanted to do. So if you put it in practice, it is very effective. Really, one of those major public health interventions that can be put into practice. Classical example of genomic medicine, the people in practice. To ensure the benefit of science reaches everybody in the society is very important. Why am I saying so much about it? 50 years or over 50 years, it's been in practice in the West. Already, most of the countries know in the respective states in the country which condition is more common, which is less common. Depending on its prevalence, they already have tests which are done when a baby is born. It's mandatory. Okay? And in our country, we don't have anything mandated. If by chance you are lucky, you go to a neonatologist who identifies that it's an inborn error of metabolism, the child gets quickly tested. If they are aware, then the child may be put onto the right treatment. But for all practical purposes in the country, it is not mandatory, it is not in practice. So although it is such a beautiful proactive way of medicine and not a reactive approach of treating after the disease has developed, we are nowhere. Okay? And NDS is not mandatory. These are textbook stuff, right? You will see lots of conditions like these. Those of you, I'm sure Dr. Saab would know a lot from the clinic, you would see so many of these who are so preventable, okay? And yet we don't do anything. So some years ago, maybe about 6 years, 7 years ago, we got a major project to screen 200,000 newborns along with 26 collaborators, with Seema Kapoor being one of the main clinical collaborator across 22 hospitals in Delhi state. And we looked at 35 easily treatable conditions for which testing was done, and approximately, of course, G6PD deficiency turned out to be the most common, but there were a few other conditions which I'm going to show you, which were so common, so avoidable, and yet we don't do anything. We did a cost-benefit analysis, huge benefit if the intervention is provided in time. It's a classical example of translational medicine and technology for the masses, okay? And we are trying, so this is what happens. Congenital hypothyroidism, according to the data that we've generated, 1 in 1,900 newborn babies in North India. And I'm saying that because our study was based in Delhi. So 1 in 1,900 babies was positive for this. All that it required was just thyroxine treatment. But the diagnosis had to be done, confirmatory diagnosis has to be in place, and the treatment has to start within 2 weeks of the baby being born. If it doesn't happen, this is what is the result. Yeah, first born in the family. You can ask, why are you putting these pictures? Why not Indian pictures? 6 years down the line, I still don't have an Indian family where I have a baby who's been treated in time to get a normal picture like this. So you will have to take it from somewhere. So here in the wheelchair is one baby, the mental retardation autism is the The second with the same mutation, but given the right intervention, is a normal child. Of course, what you need to remember is the treatment is for life. Okay? We don't know 10 years down the line whether CRISPR-Cas technology will get thing to happen, but of its own, which is a different thing. And another really sad state is this. Okay? All of you recognize. Okay? We jeer at them. We treat them so bad. Every red light, you have these hijras standing. All that happens there is one mutation which is present in 2 copies because both the parents passed it on to them. They have ambiguous genitalia, and there are other features which are typical characteristic. If they don't get the intervention, again, it is just one defect in one gene, CYP21A. If you gave a 7 rupees tablet per day the rest of the life of that particular individual, you would never be able to distinguish the person from any of us. Okay, and yet this happens to be a community which is so, I mean, it is still growing. Nothing is really being done about it. You'd say literally, when I came to Delhi, this is what they say, that hijras hospital mein aate hain and bachcho ko uta ke le jate hain. And we never understood how can a parent or how can a hospital authority actually allow a child to be taken off. Now we know. They come, see the ambiguous genitalia, and they recognize it's one of their kind, and they pick the baby and go away. Doctors know they can't do anything about it. Parents say, "ठीक है, मेरा burden चला जाएगा." So that's how their community grows. But we are where we are in that state. So this is something which is where, and you want to know the numbers? 1 in 9,000. I don't have data for South India yet, but I'm almost sure in South India the number is a little higher. Okay, so there are north and south differences because of consanguinity and other things that we talked about, but these are interventions which are possible, which should be actually looked at. So, based on our study, already Delhi government has started a program of NEED, again at the Mansi Hospital where we had the primary collaborator, which is neonatal early vision, and we are hoping and they're not doing all the conditions, they're just doing the CH and CAH, that's congenital hypothyroidism and congenital adrenal hyperplasia, the two conditions that I talked about, where they are looking at every newborn from all government hospitals, which are going to be screened, intervention provided. Okay, and we are really hoping that someday that it will become a policy decision to make it mandatory. But for something to be made mandatory, you need to have data. So first of all, you need to have data which is generated pan-India. Depending on the state, depending on the prevalence of the condition, you'll have to say, okay, in this population, this is what is common and you should be able to do. So generation of the data is most important at this point of time, but simultaneously, it's also important that the government takes a step, and this is a classical example of data to policy research. Okay, it's not just that you're doing an intervention, it's not just that you're generating epidemiological data, but you're actually able to provide intervention. So it costs us less than 1,000 rupees to do the 35 tests, because all that you require is 2 drops of blood, a set of enzymatic tests for 5 treatable conditions that we had looked at, biotinidase, galactosemia, G6PD, and the 2 other conditions that I talked about, and another 30, which is the metabolic profiling using the LCMS. And it cost us less than 1,000 rupees. But that's a project mode. Even if there's a profit margin, you know that this intervention is going to be so valuable, and we really hope, if not other genetic diseases, at least things like these will be in practice. Finally, I like to close by saying all this is good, very good. All genetic testing is important, discovery is important, etc. But each one of us should also be aware of the ethical, legal, and the social issues in human genetics or in biomedical genomics, because there are big time difficulties with insurance companies, with employers, because you are looked at as a liability if they know that you have a disease condition or you have a mutation. It may be an early onset disease or it may be a late onset disease. Your insurance will charge you a much higher premium, and so on. So these are things which you have to do. Another big problem is fetal sexing, which has come under some control in the country because of some regulations which the Ministry of Health and Family Welfare has brought into practice, but we're still not there. Ultimately, for genetic testing, for participation in any of these things, remember, it's human rights. If I say I don't want to get tested, I don't want my baby to be terminated, the fetus to be terminated, I don't want anything done with the family, the individual, the mother, the father, the family has the ultimate right to decide. So nobody else can take away that, but in the interest of the society, in the interest of health for all, it will be extremely important if they actually put this. But primary thing would be bringing about public awareness. Getting public awareness is the most important. Even if you just go and spread the message in your own community, that much more can be achieved. Okay, I like to close. All the research that I've shared with you are efforts of lots of students in the lab. I'm not even going to tell everyone's name, and a whole lot of collaborators across different hospitals from different disease conditions and funding that I've been lucky to get to be able to do that. And that's my message as always. My opening slide and closing slide never change, except the date and the time of the talk. But because I really think that's what I've witnessed over the 30 years, that we have so much more to achieve. Thank you very much for giving this opportunity. I'm really grateful that I've been able to kind of convey to you what passionate science can do for people. And Professor Raman introduced Professor Ray-Chaudhuri as somebody who was a great mentor, a great teacher, okay, and somebody who actually started genetics. But in those days, they did not have a chance to do human genetics in such a big way. So I still remember the classical picture of sex chromosome evolution between Boidae, Colubridae, Elapidae, and Viperidae, which I think was Lalji Singh's paper with Professor Ray-Chaudhuri. Such a beautiful way of showing how vipers, which is the most highly evolved group, had sex chromosomes, the ZZ and the ZW, whereas the Boidae and Colubridae didn't have sex chromosomes. So they were able to do really very good science because they asked very good questions. Today, we are blessed with the technology, and therefore, I think it's very important that if we are convinced that we can do something for the society, go all out to do that. Thank you very much once again. I'll give them this time. Yeah, I don't know. It's okay.
It's such a wonderful talk, ma'am. And traditionally, we don't take questions, but because you have designed your talk for students and you have really beautifully delivered such a complicated topic, which I was thinking is going to be so many graphs and westerns and data, but it has been so nicely.
Thanks to Professor Dakoti and Professor Raman to tell me, oh no, you can't do that.
I really got this message.
I hope you got it all.
Yes. So
Yes?
if the audience have any questions, we will be happy to take your questions.
Yes?
अच्छा मैम स्टॉप शेयरिंग on the screen screen बंद कर दें जी कर दें yes
You can please put on the lights.
put on the light.
Hello, ma'am. Such a nice talk. Ma'am, how monoclonal antibody used as gene therapy and how it's work? What's the mechanism? Can you explain?
Okay. It's a very broad question, but all that I would say is that, if you recall the example that I showed you for rheumatoid arthritis, it was a monoclonal antibody against that particular protein that we use. Okay? So all that it was going to do is block that particular protein, because the monoclonal antibody will just block the function of that protein, and that's how you will have the intervention which is coming. That's true for cancer, that's true for any of these conditions where they are using monoclonal. But for diagnosis, if you're using antibodies, polyclonal is much better. But for therapeutics, monoclonal is a better option. It's simply, all that it does is it will just do the blocking exercise. I don't want expression of a particular protein in a particular cell, in a tissue. I'm just blocking that. That's the way it works. Yeah? Yeah. Okay. Yeah. Yeah. Okay. Yeah. So so determination of Prakriti is actually Ayurveda-wide or Ayurveda physicians' forte. The classical ones, the very well-trained ones, they will start with a Nadi Pariksha. Okay, they will do the pulse examination. They already know part of it. They will look at you as an individual. They will look at a multiple phenotypic traits. Okay, and they will characterize. They will ask you what you eat. They will ask you about your habits. They will ask you a whole lot of questions which you can answer for yourself, and they will determine the prakriti based on that, which is what an Ayurveda physician does. But if we have to use it in a research mode, we have to have a much more objective way. And therefore, CCRAS, that's under the Ministry of Ayush, we have the Center for Ayurveda Research, where they have already come up with a questionnaire which has got about 55 questions, which is administered to all willing subjects who want to participate in the research. And after they do that, you still have to have an Ayurveda physician validating that with the person right in front of him. So filling up the questionnaire is one part, and actual validation by an experienced physician is another part. So they have revised the questionnaire in the last 5-6 years. So most of us who do use that questionnaire, but we are very, very particular that we have a very good Ayurveda physician on board to make sure. And there is always consensus diagnosis with the second one. And if you're wanting to go through with this mode, there is really no marker at this point based on the prakriti. So biomarker discovery for the prakriti groups. Okay, when I say that, genetic markers, which Dr. Pangaraj's group has come with something. There are a couple of groups in the country who are doing it, but at the moment, if you ask, are there any biomarkers which I can just use to say this is Vata, Pitta and the Kapha? We are not there, but a lot of effort is going into that to identify biomarkers. So questionnaire is what is available to you today. Biomarker discovery is something which is being attempted, they're trying to bring out these biomarkers to go ahead with the prakriti determination. Yes. Yeah, but that is for... Okay. No, that is, when you're talking, you want to kind of look at a drug which is being used in Ayurveda. You want to see what does a drug do, okay? How does a drug work? I already told you very early on in my presentation, there are some things that you can do with animal models. There are other things that you can't do. A prakriti determination in animals is out of question. I need human subjects. But testing a drug molecule, seeing the way the drug is working, or a particular phenotype. I make a Parkinson's disease model with Drosophila, let's say so. I certainly can screen a whole lot of drugs which are being used by the Ayurveda clinic in the animal model. That's how you will use animal models for doing drug testing or understanding some of the phenomena. But for using human subjects, you have to have the prakriti determination.
I think we should stop here, and thank you all for asking questions. Thank you. Thank you, ma'am. Thank you once again. Very interesting talk, and you have really walked us from a gene to a possible treatment target with your own research. Thank you again. So with that, I would like to invite our guest, Professor Shyam Sundar. Sir, please present a memento to them. Please present memento. Sir, done. Thank you so much.
स्टेज पे तो आराम से घूम रहा था कैमरा.
So I also request you, because you are the sir is presiding over the function, so I would request you for your remarks.
Thank you for the invitation. I'm really moved by the invitation to this wonderful lecture. And there are I mean, what was striking for me was really your connection of genetics to Ayurveda. They're really very, very fascinating. And I think if you have you in the Institute of Medical Sciences, we have Ayurvedic faculty, big faculty, and interact with them, it will be really great. So I hope we have Dr. Yamini Bhushan here, who is from Ayurveda faculty. So he could really facilitate her one more visit to the Ayurveda or medical sciences. That would be great. Another thing which I wanted to tell you that to see this hall full of young budding scientists is really very pleasing. And I'm really very happy to see that people are standing, that there are no seats, all seats are taken. And the lecture or oration is definitely, fascinates or pleases the awardee. But I think your lecture has probably excited many, many young people and colleagues, younger colleagues, to do the research in the way you have done. So once again, I thank you very much for coming here. And I thank Dr. Rahman and his team who really got this lecture organized at the earliest moment, COVID permitting. Thank you very much.
Thank you, sir. Now we are almost towards the end. So now I would like to invite Dr. Gaurav Pandey. Dr. Gaurav Pandey to propose vote of thanks. Sorry, I forgot. See, Gaurav, please.
Good evening, everyone. On the behalf of Professor S.P. Ray-Chaudhuri Memorial Foundation, I stand here to express my vote of thanks. A special thanks to our chief guest, Professor Shyam Sundar, IMS BHU, for accepting our invitation and for being with us here today. On the behalf of the present and the past members of Foundation and the Department of Zoology BHU, I extend my heartfelt gratitude to today's speaker, Professor B.K. Thelma. It was a privilege to hear your lecture, especially on the approaches and the variation and the information which lies in the Indian population and how it can be applied to formulating new precision drugs which can be more effective than the existing ones. So thanks. I thank our head, Professor S.K. Trigun, for all his support in organizing this event. Our sincere thanks to all the esteemed invitees for their gracious presence, Professor Anil Tripathi, Director, Institute of Science, senior professors, faculty members of various departments. I also thank many invitees whom you cannot see because they have joined within India and abroad via Google Meet. And also this meeting is being live telecasted on YouTube. So I thank everyone who has joined via online mode. This lecture would not have been possible without the constant encouragement, advice, and contribution of senior members of the Foundation, Professor S.C. Lakhotia, Professor B.N. Singh, Professor Mercy Raman, Professor Rajiv Raman, Professor Jagat Roy, and Professor Madhu Tapadia. I also take this opportunity to thank all the outgoing members of the governing council of this foundation. Finally, I would like to extend my sincere thanks to all the students and staff members who have actively participated in this event with lots of energy and enthusiasm. I'm sure, and it's actually not possible to take names of all of the people who are associated with this meeting. So I would like to thank everyone who was associated in any form with today's lecture. Your contribution is highly appreciated. In the end, I thank all the audience for gathering here and attending today's lecture. So you all are requested to please join for refreshment. Thank you.
लेट चल रहा है ना पीछे लाइव ही रहना बस रिकॉर्डिंग नहीं है इसकी ना
नहीं रिकॉर्डिंग नहीं सिर्फ स्ट्रीम